Okay, so before starting this section, macromolecules, section number 3 for lipids, you should have completed polymerization and also carbohydrates. You should have also covered functional groups. You should have also covered organic, because all of these macromolecules are organic. Okay, so lipids. So lipid is kind of an amorphous term, so I'm defining right here. The polymer, lipid is kind of that vague term, and a specific polymer that we're going to be talking about is the triglyceride. Another specific one we're going to be talking about is the phospholipid. So those are our polymers made of many parts. The monomers of every single one of these is the fatty acid. So as the name implies, and as I told you in the functional groups section, it has an acid group in it. So what the fatty acids look like is they have an even number of carbons. They typically have more than 10 carbons in this chain, and at the very end they have an acid group. So 1, 2, 3, 4. So here's 20 carbons long, and here's our acid group on the end. And remember, this acid group could also be written COOH, COO-, or it could also be written as this, with a minus charge, because this H will go off into solution, and that's what makes it an acid, because it'll increase the concentration of H, because that just falls right off as far as we're concerned. Okay, so here's an excellent example of a fatty acid. Acid group, and this part is the fatty part. So this is a long nonpolar tail. Nonpolar tail. And it's nonpolar, if you remember, because it's got lots of C's and H's and nothing else. This part is polar because we see those electronegative groups, those O's. It's got O's in there. So O's and N's mean that something's polar. If it doesn't have O's and N's, if it's just C's and H's, it's nonpolar. So that's your reminder of that. So this is why, if you think of lipids, you can think of something like oil or butter. And if you think of the structure of water, that's polar. So our nonpolar oils do not dissolve in our polar water. And we're going to see this in a lab as well. So this macromolecule is pretty much the only nonpolar molecule that we're going to concern ourselves with. So this particular fatty acid is saturated. If you've heard of saturated fats, they would look like this. So that means they have no double bonds. Whereas an unsaturated would have double bonds. It would not have triple bonds. So we're going to see some double bonds in here. And there's a couple different ways to draw it. So here's a double bond. So here's an unsaturated fatty acid. And then double bonds in fatty acids can also look like this. And they can occur anywhere in the chain. So all of these three are all fatty acids. Double bonds or not, it is still a fatty acid. And it can have multiple double bonds, or no double bonds, or mixtures of ones that look like this and ones that look like that. Anywhere on this chain, it doesn't matter. These are all fatty acids. And you can tell fatty acids by the long hydrocarbon chain and the acid group, the acid functional group. Okay, so now let's talk about polymerization. So we're going to talk mostly about the triglyceride, and then we'll move on to the phospholipid, and then we'll be done. So this triglyceride, tri means 3. This glycer refers to a glycerol. So a glycerol is an alcohol sugar that looks like this. So this is glycerol. And this is the backbone for polymerization of the fatty acids. So here's our fatty acid, big long chain. And you can see there's 3 OH groups, so there's spots for 3 fatty acids. Okay, and each one of these is going to combine like so. And we'll practice doing this in class. And it doesn't matter if this O gets circled or that O gets circled, all of these are equally correct. So as long as what's in the circle has the formula H2O, you are doing this perfectly correctly. So when this combines, it loses 3 total water molecules, 1, 2, 3, 1 for each, because we're combining 4 parts. This is part 1, this is part 2, this is part 3, this is part 4. So 4 monomers are coming together, and we release 3 H2Os, and that's going to make our triglyceride. So that bond looks a little different than in the carbohydrates, but you can still tell that it's a triglyceride if you look at how many fatty acids there are. So we see now 1, 2, 3 long chains. And if we compare this to the fatty acids up here, these aren't connected to anything. They still have this acid group that's not connected to anything, and they only have 1 long tail. So here we can now see the acid group is now connected to something. We have 1, 2, 3 tails all connected to this E-looking thing. So this is how you're going to identify a triglyceride. So it has more than 1 fatty acid. So this is much bigger than a fatty acid. And this kind of shields this polar part. And this makes the whole thing pretty much nonpolar. And this is how you store your fats. There's one other type of fatty acid that we're going to talk, or lipid that we're talking about, and that's a phospholipid. So I'm not going to draw how those are made. I'm just going to draw the result. So this is from 2 fatty acids and a phosphate group. And we see this charge on these oxygens. This part is really, really, really polar. And this part is really, really, really nonpolar. And so we're going to see this in the membrane. So this is a phospholipid. So it's named phospholipid because it's got a phosphate head. This is the polar head. And it's got a nonpolar tail. And you'll notice that there's 2 tails because the glycerol had 3 total connections that we could have made. 1, 2, 3. One of those connections is connected to a phosphate group. So that's how a phospholipid differs from a triglyceride. Also, the triglyceride is almost entirely nonpolar. This has a very polar head and a nonpolar tail. This is extremely important in membranes, and we'll see this again later. And it's very important that this only has 1, 2 tails. And so we'll come back to this structure again in the next chapter.